My dear student friends, in today's lecture, let us study the predisposing factors for diseases of silkworms. Sericulture, the highly enterprising occupation of raising silkworm larvae with the aim of producing silk cocoons for ultimate production of silk, is often marred by the disease incidence in the silkworm colonies. The diseases such as bacteriosis, virosis, flachere, muscardine and pebrine are caused by different bacteria, viruses, fungus and microsporidia. These microbial agents are ubiquitous in the rearing environment and sometimes attain epizootic proportions, taking advantage of certain conditions during the course of silkworm rearing. These factors and conditions are termed as predisposing factors in causing the diseases. These factors act in different ways, one by promoting the microbial growth and another by weakening the host larvae, making it susceptible to the pathogens. The common factors found such as temperature and humidity conditions have direct bearing by way of stressors and indirect bearing by way of influencing the quality of mulberry leaf in the success of silkworm crops. Generally, unfavorable temperature and humidity conditions, inferior mulberry foliage, inappropriate rearing houses, improper rearing practices, poor ventilation, etc. are some of the predisposing factors in causing silkworm diseases. Unfavorable conditions or stress factors weaken the insects and make them prone to pathogen infection. Similarly, conducive environmental conditions may favor the easy multiplication and pathogen build up in the rearing environment. Once pathogen gets entry into the host and conditions favor its multiplication in the host, disease is manifested in the host. For better understanding, the topic is being explained into different subheadings and they are: 1. Disease development, 2. Improper rearing houses, 3. Faulty rearing practices, 4. Improper disinfection and hygiene, 5. Nutrition and host susceptibility, 6. Environment and host susceptibility, 7. Synergistic association of pathogens, 8. Host population density and 9. Alternate host. First, let us study the disease development in silkworms. The course of infection can be separated into different periods or stages: incubation period, the beginning of the disease and the height of disease and the termination of disease. Incubation period is the period from the entrance or introduction of pathogen into the host body until the development of symptoms or signs. The beginning of the disease is the period from the first appearance of sign or symptoms until the disease is fully manifested. The height of the disease is the period from full attainment of sign or symptoms until its beginning to fall or reach a steady state. It is during the period of height of disease the microorganism is most active, the sign or symptoms are more severe, lesions produced will be greatest and the toxin, if any produced, will be in maximum amount. The termination of the disease is the end of disease condition. It may result in the death or recovery due to immune response. The death may be due to lack of immune response or low level of response. The second aspect of our study is improper rearing houses. Rearing houses are the place where silkworms are reared. In India, farmers are found to rear silkworms in their dwelling places. It is common sight that the farmers keep the worms by the side of their kitchen and sleep by the side of the larvae they rear. But in such conditions, a thorough disinfection may not be possible. Much care is required in providing separate rearing house for hygienic rearing of silkworms. The rearing house should be made in such a way that proper air current and ventilation is ensured. rearing houses should be made in such a way that proper disinfection could be carried out. Crevices or cracks in the floor and walls should be avoided as there is chance of pathogens getting accumulated. This in turn can turn into a reservoir of pathogens resulting in secondary contamination. Ventilation of the rearing house is essential for natural movement of fresh air into the rearing environment and contaminated or heavier air out of rearing house. Cross ventilation is essential for good air movement within the rearing house. Improper construction or maintenance of rearing house aids in the build-up of pathogens and causes a reason for disease occurrence in the silkworm colonies. The third aspect of our study is faulty rearing practices. Practice of proper rearing practices are essential for maintaining good health of silkworms. Providing timely feed with a good mulberry in sufficient quantity, proper molting care, etc. are important. Larval density in the rearing bed increases the incidence of grassery and other silkworm diseases. Crowding as a stress has been used to bring down latent infections in the insect colonies. Therefore, maintenance of optimum larval density in the rearing bed is essential for the healthy growth of silkworms. During molting, the larvae should be provided with good air currents and feeding should not be given while they are under molt. It is observed that increase in carbon dioxide contents in the air of rearing room affects the health of silkworms and leads to induction of NPV. Carbon dioxide emitted from silkworm bodies, mulberry leaves, burning of fuels, etc. possibly lowers the pH of hemolymph and stress on the silkworm. Similarly, the air current has a great effect on the physiology of worms, especially when there is high temperature and high humidity. The air current brings fresh air into room and reduces the body temperature of silkworms. This helps in improving the health of silkworms and cocoon crops. Therefore, it is important to maintain optimum larval density and allow fresh air circulation during the silkworm rearing. The fourth aspect of our study is improper disinfection and hygiene. Disinfection is the process by which the pathogens are eliminated in the rearing environment. This is vital for the success of silkworm crops. Improper disinfection of rearing house and environment causes the buildup of pathogens in the rearing environment, and this can be destabilizing factor. If the pathogen load goes beyond a certain level, silkworms become susceptible to their infection, and further multiplication of the pathogens in the silkworms will affect the entire colony, and this result in the disease spread in the environment. Similarly, rearing hygiene during the course of rearing is vital, which includes the identification and elimination of diseased worms from the rearing bed, dusting of bed disinfectants, maintaining of personal hygiene, etc. Improper disposal of diseased larvae, bed refuse and rearing waste is a reason for pathogen buildup in the environment, and this in turn will affect the other silkworm crops in the vicinity and future crops also. The fifth aspect of our study, nutrition and host susceptibility. First, let us see the poor quality of leaf. The growth, development and cocoon production are greatly influenced by the quality of mulberry leaf in which silkworms feed. Silkworms grow at an optimal rate only when fed with mulberry leaves in good physiological condition. Late age worms require mulberry leaves containing moderate content of moisture, high crude protein, starch and soluble carbohydrates. Silkworms fed with mulberry leaves containing low protein, sucrose or high cellulose becomes more susceptible to viral infections. The silkworms fed on artificial diets containing powdered mulberry leaves from autumn were comparatively more susceptible to viral diseases than the ones fed on artificial diets containing powdered mulberry leaves of spring. In Japan, the viral diseases are low during spring, but increases in autumn, which is attributed to poor quality of mulberry leaves during autumn. Lack of suitable quality food stimulates the latent viruses into active form. Prolonged storage of leaves leads to loss of leaf quality and affect the palatability of silkworms, leading to malnutrition and making them prone to various diseases. Susceptibility of the viral diseases is directly proportional to the rate of deterioration of mulberry leaf quality associated with prolonged leaf preservation. The poor quality mulberry containing low protein, sucrose or high cellulose makes silkworms comparatively more susceptible to infection by pathogens. The susceptibility of pathogens to bacterial infection is governed by the nutritive factors. The silkworms also derive precursors of antibacterial and antiviral substances from the mulberry. In silkworms, the caffeic acid derived from chlorogenic acid from the mulberry leaves is converted in the silkworm's digestive tract into antibacterial caffeoquinone. The fecus of silkworms is also known to contain two antibacterial phenolic acids, namely procatechuic acid and p-hydroxybenzoic acid, which must have been derived from mulberry. Terpenoids, particularly B-carbophyllene, myrcene and pinene, phenols and flavonoids including catechin and tannins in plants are also known to inhibit the growth of microorganisms in insects. Food quality and starvation. Similarly, starvation is unfavorable for silkworms at any larval stage. Prolonged starvation may have been predisposing factor for grassery. Poor nutrient availability becomes a major factor in higher incidence of viral diseases. The sixth aspect of our study is environment and host susceptibility. Ecological factors influence the epizootics by way of acting either on the virus or on the host. For example, rearing at a constant temperature appears to make the silkworms quite resistant to viral infections, while warm weather coupled with higher humidity seems to be a cause for viral epizootics. Significant correlations between the incidence of grassery and temperature have been recorded. The high rate of nuclear polyhedrosis in a colony of silkworms during summer is attributed to lowering of incubation period of pathogen and a higher rate of multiplication of BMNPV at a temperature greater than the optimum silkworm rearing temperature. Similarly, sudden fluctuations in temperature and humidity induced grassery in silkworms. Pathogen and environment interaction is an important factor in the causation of diseases in silkworms. High temperature and low humidity conditions are attributed to higher incidence of grassery during summer months. Fluctuation between day and night temperatures and relative humidity prevailing in the rearing house were observed to be major cause of high incidence of grassery. High temperature and low humidity are stressors which predisposes the grassery disease in silkworm colonies. Temperature and humidity have a direct influence on incidence of viral diseases, and these factors are coaxial in their occurrence. Therefore, it is difficult to account for individual factor effects. The least incidence of grassery has been observed under the optimum conditions of temperature, that is 25 degrees centigrade and humidity of 75%, while increase or decrease in these factors increase the incidence of grassery even in uninfected batches. It shows that silkworms may have subacute levels of pathogens which under optimum conditions of environment may remain latent. But when the silkworms experience the stress due to extreme conditions of temperature, humidity and other factors, it may lose its natural resistance and the latent pathogen may start multiplying suddenly, leading to epizootic conditions. This may be one of the reasons for high incidence of grassery during rainy and summer seasons. It has also been observed that the incidence of NPV is higher in sudden changes from room temperature to low temperature of 5 degrees centigrade than in gradual change of temperature. It indicates that heat and cold treatments may disturb the physiological condition of silkworms, which in turn may cause the transformation of virus from non-infectious state to an infectious state. Similarly, the relationship of humidity with NPV induction indicated that 100% relative humidity caused in higher grassery level and the rate of induction gradually decreased up to 50% relative humidity and then further increased with the decrease in RH, indicating the role of RH in inducing the disease in silkworm colonies. Therefore, decrease or increase of temperature and humidity alters the NPV incidence. Possibly the changes beyond the certain levels exert metabolic stress on cellular level. Such stressful conditions might affect the host defense system against the viruses. The rise in silkworm rearing temperature accelerates the development of bacterial diseases also. The silkworms exposed to small doses of Bacillus thuringiensis variety LST leads to chronic infections, but becomes acute and rapidly develops fatal bacteriosis after a short thermal shock of 30 degrees centigrade. High temperature and humidity leads to dysfunction of alimentary canal, which in turn leads to enterococcus multiplication. The integument in insects is a strong barrier for many microbes such as viruses, bacteria and fungal spores. Among the various environmental factors, humidity and temperature are to a lesser extent light and air movement play important role in affecting infection and sporulation of endomopathogenic fungi in silkworms. When high humidity conditions persist, the fungal spores germinate and penetrate the integument to cause mycosis in silkworms. The relative humidity of 90 to 100% is most favorable for the germination of the conidia. The humidity lower than 70% is unfavorable for germination of conidia. Similarly, the conidia germinates optimally at a temperature range of 26 to 28 degrees centigrade under favorable humidity conditions. At temperatures above 33 degrees centigrade, the conidia do not germinate. Beauveria bassiana and Beauveria brongniarti grows luxuriantly at 20 degrees centigrade and are active between 16 degree to 30 degrees centigrade. The temperature 20 degrees centigrade for Aspergillus tamari and 30 to 35 degrees centigrade for Aspergillus flavus and humidity of 85 to 95% predisposes the development of aspergillosis in silkworm colonies, especially in young silkworms. The fungal conidia can survive for over 1 to 5 years depending on the temperature and humidity in the environment. While developing a package for managing silkworm diseases, these factors are to be taken into consideration. The seventh aspect of our study is synergistic association of pathogens. Synergistic association of pathogens predisposes the silkworms for getting severely affected and is a cause for great loss. The involvement of different coccus and bacillus species bacteria along with a picornovirus, the BMIFE causes Thate The Streptococcus faecalis variety liquefaciens, Staphylococcus aureus and Staphylococcus epidermidis individually cause a low level of pathogenicity in silkworms, but in combination with the BMIFE, the mortality level goes high. Bacillus species of bacteria isolated from Thate infected larvae were non-pathogenic individually, but in combination with BMIFE, they become pathogenic, causing high mortality with the symptoms of flashery. The existence of synergistic action of Streptococcus faecalis and Serratia marcescens with BMIFV and DNV in causation of higher level of mortality is established. Streptococcus species, Staphylococcus species and Serratia marcescens with BMIFV and DNV were reported to shorten the incubation period, leading to higher level of mortality in shorter period compared to that due to individual infection by these pathogens. BMCPV in association with BMIFV and bacteria enhance the disease level in silkworms. Though the different pathogens multiply in different parts of the body and cell, the host succumbs to infection in shorter period. Disease incidence may be due to lowering of gut pH by the infection of bacteria, which provides congeniality for the growth of other pathogens, allows fast multiplication and could lead to degeneration of peritrophic membrane, which blocks the absorption of nutrients, and this is reflected in the cessation of feeding, sluggishness, etc. A fall in the gut pH due to bacterial infection has been reported by Ono and Cato in 1968. A change in blood pH also leads to general paralysis in silkworms due to leakage of gut contents into the hemocoel. Higher concentration of pathogens cause more virulence. The infection process is determined by the balance between the device of aggression of the pathogen and its virulence, the mechanism of defense of the insect host, its genetic immunity and the stress exerted by the environment or other agents. Host population density is the eighth aspect of our study. It has been generally observed that natural epizootics of viral diseases are more common in situations where there is a high host density or overcrowding. Stress produced by overcrowding has been reported to increase the susceptibility of insects to diseases. Significant incidence of grassery was observed in the rearing trays as the larval density increased. The change in the microclimate in the rearing areas due to overcrowding affects the physiological functions concurred with the induction of BMNPV. The population density of larvae reared has a direct effect on the incidence of grassery. Therefore, it is important to maintain optimum larval density during silkworm rearing to obtain higher rate of effective rate of rearing. The NPV of silkworm often remain in latent condition and gets activated under stressful conditions of host, and apparently disease bursts out later in epidemic proportion despite of best efforts such as disinfection and good conditions of rearing. The ninth and final aspect of our study is alternate hosts. Alternate hosts are reserves of silkworm pathogens which, when contaminates the mulberry foliage with the disease causing pathogens, the disease attains an epizootic condition. Many insect pests and mulberry are observed to be infected with silkworm pathogens. Natural infection with Nosema bombycis are observed in Pyrus rape, Spodoptera diparvata, Spodoptera exigua, Spodoptera littera, Tricolopusia knee, Diaphania pulverularis, etc. Similarly, BM NPV infects insects such as Limantria dispar japonica and Dendrolimus spectabilis. BM CPV infection is observed in fall webworm Hyphantia cunea and Spodoptera litura. BM CPV also infects Dendrolimus spectabilis, Philosamia cynthia prarai, Malacosoma hustrata, Callias diurotheme and Theophila mandarina. The mulberry pyralid Glyphotes pylolis is a major alternative host to BM DNV1 and BM DNV2. Vivaria bassiana and other fungal pathogens occur worldwide, which has largest host list and occurs in soil as ubiquitous saprophytes. These insects in the vicinity of mulberry garden causes high buildup of pathogens in the environment and can lead to epizootic conditions in the silkworm colony. Thus, the topic can be summarized that the silkworm rearing has to be conducted in a scientific and systematic manner. As silkworms are prone to many diseases and many pathogens are involved in causing these diseases, the process has to be handled appropriately. There are many predisposing factors which aids in the disease infection, manifestation and loss of crops. The main predisposing factors are pathogen buildup in the rearing environment, conducive factors for pathogen germination or development, weakening of the physiology of the host larvae, improper rearing conditions, adverse environmental conditions, inferior or improper feed, etc. General precautionary measures are emphasized to rear silkworms under optimum conditions of temperature and humidity, and supply of nutritious food in order to enhance the host's natural defense system against pathogens. By strictly following these measures, silkworms can be protected from diseases. Further, to overcome some of these factors, there are specific strategies and each factor has to be carefully dealt to avoid infection to the crops for successful silkworm harvest. Thank you.